All right. Hello and good afternoon, everybody. This is our third Fireside Chat in the series, coming to you from our home offices and kitchen tables. Today, we're going to be hearing from, again, faculty researchers in the UGA College of Veterinary Medicine, and we're going to be discussing vaccines and therapeutics to target SARS-CoV-2. We're building on chat number one, which was related to transmission and diagnostics. Chat number two, which was the immunological response that various people or patients are having to the infection. And now we're going to talk about how to prevent and treat the infection. So we have a full agenda of things to talk about. So we're going to kind of jump right in. I'm joined again by Chet Joyner and Karen Norris, who you heard from last week. And we also have Biao He, as well as Jared Musa joining us on the panel as well. And again, we'll kind of follow the progression of introducing people more fully as we get to the various sections of their expertise. So I'm actually going to start the session with a very high-level overview of the different types of vaccines that are actually being developed to target the SARS-CoV-2 infection. So we'll first start with the actual virus vaccines, and there are two types. They're a weakened virus and an inactivated virus. To my knowledge, I don't know that there are any vaccines that are being developed using the weakened virus. However, you all probably are familiar with some of the versions of weakened virus vaccines if you've had the measles, mumps, and rubella vaccine, or if you've had the yellow fever vaccine for traveling to tropical countries. On the inactivated virus side, there are at least two companies that are developing SARS-CoV-2 vaccines using this methodology. It's basically a virus that's chemically inactivated, injected, and induces an immune response. And so you're probably also familiar with inactivated viruses in your lifetime if you've received the rabies virus, or the seasonal flu vaccine is also an inactivated virus. Moving on, there are nucleic acid vaccines that are actually being developed. There's a DNA vaccine that's actually moving into phase 1 clinical studies. Basically, it uses a piece of the DNA genetic material from the spike protein and is delivered via vaccine to also induce an immune response by allowing cells to produce the protein that was encoded in that DNA. And on the RNA side, this is a vaccine that's probably gotten the most attention. This is Moderna's vaccine. So they are actually taking a piece of the RNA genetic material that encodes the spike, and they are delivering it in a proprietary nanoparticle so that the spike protein will be made in your cells and then presented to your immune system to induce an immune response. And so a couple positives and a couple things to think about for nucleic acid vaccines. There are no nucleic acid vaccines that have been approved for any pathogens to date. But a positive is that the manufacture of these vaccines can be scaled up pretty quickly. So I'm going to move on to the last two vaccines that you can use or platforms that you can use to develop vaccines. And interestingly, these two platforms are actually being used at the University of Georgia to develop vaccines to SARS-CoV-2. So the first are viral vector vaccines. There are two flavors. There's a replicating flavor and a non-replicating flavor. In these particular vaccines, you use a virus, an inactivated virus. Some of you may have heard adenovirus being talked about in the media. So an example of a non-replicating viral vector based on adenovirus is the virus that's being, or the vaccine that's being developed by Johnson & Johnson and also by the University of Oxford. Again, you use this vector backbone to introduce the genetic material from the spike gene, and you deliver these vector virus vaccines into patients, allow the development of that piece of genetic material, spike protein, and that induces an immune response. In a few minutes, Biao He is actually going to talk about his viral vector vaccine, which is a replicating viral vector vaccine. There have been two viruses in the last 2 years or so that have been approved that are viral vector vaccines in the U.S. One targets Ebola and one targets dengue fever. Interestingly, Biao has been working on a coronavirus, on his coronavirus vaccine for a little while now. It didn't target SARS-CoV-2, but it targeted another member of the coronavirus family, which is the MERS coronavirus. And like I said, in a few minutes, he will walk you through his research program. And then the last or fourth type of vaccine being developed for SARS-CoV-2 are protein-based vaccines. And they can take two flavors. One, you actually deliver the protein that is the spike protein in an emulsification directly to patients. This is actually a fairly common type of virus. So if any of you have gotten the hepatitis B vaccine, this is an example of a protein-based vaccine. Or if you've gotten the shingles vaccine, also a protein-based vaccine. And then there's a second flavor for protein-based vaccines that use something called viral-like particles. So it takes an empty virus, and then you conjugate or you attach in this case, the spike protein to that empty virus, and then that is delivered via an injection. And so this viral-like particle type of vaccine is actually what Ted Ross is developing at the University of Georgia. And there are a few other faculty, Dan Perez in particular, has a significant amount of experience in developing the virus-based vaccines. So I'm going to move on now and just give you a view of the number of vaccines that are actually in various forms of development. So last count, there's over 150 vaccines in development across the world. As you can see, there are the most in the exploratory phase. This is actually where Ted Ross's vaccine is. And then you can see that it funnels down into just a handful of vaccines in phase 1. Biao He's vaccine is actually in this preclinical phase. and then pointing out that there are probably about 6 vaccines in total that are in this phase 1 stage. So we really are trying to pull out all of the tools and experiences of vaccine development to target this particular virus. I will say, I didn't say this in the beginning, but I invite you all to use the Q&A section to go ahead and ask your questions. I will monitor that section, and we will do our best to address your questions as we come through each of the sections that our faculty are going to talk about their expertise. And so with that, I'd like to introduce you all to Biao He, who's going to talk about his vectored vaccine for SARS-CoV-2. He is the Fred C. Davidson Distinguished Chair in Veterinary Medicine, and he is a full professor in the Department of Infectious Diseases. Biao's research is focused on viral diseases and vaccine development for emerging infectious diseases. And so I'll let Biao talk to you a little bit about the vaccine that he's been working on.
Thank you, Michelle. Thank you for a very nice introduction. And I am happy to be here and very excited to tell you about the work we're doing on COVID-19 vaccine development. And the approach we have been using is to re-engineer an existing product. And as many of you know, if you are you have a dog and have ever had to board your dog, you have to use a Kennel Cough vaccine. So in the Kennel Cough vaccine, it contains live Parainfluenza virus 5. It's commonly known as canine Parainfluenza virus, CPI, in the field. So you can see there are many products on the market. So this is a live virus. And normally, the way we immunize it is through intranasal. So you can see here some of our first-year students doing clinic and getting dogs vaccinated with a Kennel Cough vaccine. So this is a live virus, and then this is one of the points I want to make. That is, it's a live virus. It goes into the nose, and as you know, COVID-19 is caused by a respiratory virus. So develop a vaccine through the same route of immunization gives us really an advantage over other approaches. So in the past few years, we have been working on develop a vaccine, as Michelle mentioned, for MERS. MERS is a virus, is a disease caused by coronavirus that's very closely associated with current SARS coronavirus and SARS coronavirus 2, which causes COVID-19. So in past about 5 years, I'm working with a group at the University of Iowa, and we developed a vaccine for MERS coronavirus using the viral vector that has shown early on that it's contained in the Kennel Cough vaccine. So we made this vaccine through an intranasal route, and the work actually been published in past last month. So, but the work has been going on for about 5 years. Well, one of the things I want to point out in here is that I'm very grateful for the support many of you give us, because I am fortunate to be the endowed chair for the Fred C. Davidson endowed chair. And as you know, the coronavirus was not on the radar for many of the funding agencies. To do this work, we really have to use resources we can find. And this is one of those situations that the endowment made a huge difference to allow me to do this work in the past 5 years when people were not really paying attention for coronavirus. And we developed this vaccine, and we recently published. Now, because the MERS coronavirus and SARS coronavirus 1 and 2 are very closely related, so we are able to use the same strategy to develop a vaccine for COVID-19. So here is a more detailed explanation on what we do. So we take PIV5, which is the same virus as said in the Kennel Cough vaccine. We genetically modify by insert the the spike gene that encodes the spike protein. So we will into this virus. So we make this recombinant virus now that we will now calling the CVX UGA1, because this is the vaccine we're going to a human clinical trial, hopefully by the end of the year. So we have done quite a bit of a preclinical work showing efficacy in animal models. So we're very excited and very grateful for the support we have gotten from many of you in the past. And so we're doing our best to get this thing into human as quickly as we can. So I will be happy to answer any questions you have. And I think my 5 minutes up.
So Biao, where are you exactly in development of your MERS vaccine versus the COVID-19 or SARS-CoV-2 vaccine?
So in terms of vaccine development, actually, we're quite advanced in the COVID-19. As you know, MERS vaccine, we spent about 5 years doing a lot of groundwork, essentially to figure out what's the best way to do it. And again, that's really very important. So knowing how to make a MERS vaccine, essentially, we use the same blueprint to make a COVID-19. And in the MERS situation, we have tested in 1 animal models. But in the COVID-19, we have done 2 animal models now, and this is actually satisfied the FDA's requirement in general. They need to see efficacy in 2 different animal models before they allow you, of course, to do tests in human. So this is where we're at right now, in consultation with FDA to move the vaccine into human clinical trial. And for the MERS, and again, still it is circulating mostly in the Middle East right now, and we would like to continue that work. But as you can see right now, everyone's attention and the effort and resources are focused on getting the COVID-19 into human as quickly as possible. Great.
Thank you. That's very helpful and hopefully pretty promising. So I wanted to share also that there are additional vaccines being developed that play a role in helping to prevent deaths from COVID-19. So some of you may have heard in the media that some of the patients that are having the severest symptoms and ending up on ventilators are actually developing secondary lung infections, and that is posing a significant problem as far as being able to not only clear that lung infection, but also mount an immune response to clear the COVID-19 infection. And so Karen Norris is actually joining us again to talk about her protein vaccine that targets these life-threatening invasive fungal infections. Karen is a Georgia Research Alliance eminent scholar. She's also the Charles H. Whitley Chair of Immunology and Translational Biomedical Research and a professor in the Department of Infectious Diseases, like Biao, which is in the College of Veterinary Medicine. Her research is focused on vaccine and immunotherapeutic development for various respiratory infections in immune-compromised individuals. So I'll let Karen talk about and give you an update on her particular vaccine, and then we'll move along into what are the timelines for development of vaccines.
Thank you, Michelle, and it's good to be with everyone today, and those of you who are with us for the last 2 sessions. As Michelle said, we've learned a lot in a short period of time about the course of infection with the SARS-CoV-2. And what has become apparent during this time is that there are certain populations and certain individuals that are more susceptible to developing COVID-19 and more susceptible to developing severe consequences of COVID-19. And I think we're all aware that the elderly are particularly susceptible but it has also become apparent that immunocompromised individuals, so people with weakened immune systems, such as individuals with autoimmune diseases like lupus or multiple sclerosis, individuals that are being treated for cancer or transplant recipients, all of those individuals have a somewhat weakened immune system. We've also found out in the course of looking at patients that do poorly with COVID, with the COVID infection, is that even individuals with chronic diseases like type 2 diabetes, obesity, and hypertension are more apt to develop severe consequences and poor outcomes. And we don't completely understand that. What has become clear, though, is when we look at these hospitalized patients, particularly the ones that require a ventilator and the ones that have a very poor outcome, is that they also, in addition to having the SARS virus, they develop other lung infections, as Michelle said. My lab has been working for a number of years on different viral and fungal and bacterial pathogens that affect the lungs and cause lung diseases. And we're particularly interested in understanding the immune response in these individuals with weakened immune systems and individuals with diseases like diabetes or hypertension, and why they're so susceptible to these types of infections. So what is showing on the slide, there are a group of organisms that are fungal organisms, and they're present in the environment, they're present in the air, and we're all exposed to them. And in healthy individuals, we breathe those in, as the slide is showing. If we're healthy and have a working immune system, we clear those organisms and we do fine. There are individuals that are slightly weakened in their immune systems, and they are called susceptible hosts. And so they breathe in these organisms, and they don't cause overt disease, but they are carried in their lungs for a long period of time, and they're thought to contribute to transmission. The third group is the one that we study the most, and that is immunocompromised individuals. And when they encounter these same bugs that healthy individuals do, they develop a lung infection that is very, very severe. Okay. What we've learned in the COVID-19 situation is that a high percentage of the patients that do poorly in the hospital and have poor outcomes also have these types of invasive fungal infections. And so prior to the COVID pandemic, my lab has been developing a vaccine that targets the 3 top fungal infections in these populations. And so we are working to develop that protein vaccine for these types of at-risk populations. And this would benefit the immunocompromised individuals that I talked about that are more susceptible to these types of infections, in particular COVID individuals that would be susceptible to COVID-19. And so that work is progressing, and we have that vaccine through animal models, as Biao has talked about in his, and we are in the process of getting that vaccine ready for regulatory approval at the FDA. So that is where we are in that. All of this work helps us be able to protect these individuals that are immunocompromised or that would be at particular risk for secondary infections on top of their SARS-CoV-2.
All right, I'm gonna jump in here because we already have questions coming in. So one question is, would DNA-based vaccines be ineffective for RNA viruses? And I'll open that up to Karen and Biao if you guys want to take a stab at that.
Yeah.
Well, I think the DNA vaccine, if it works, it should really work against all kinds of viruses. That doesn't matter is DNA virus or RNA virus, because the way DNA vaccine works is that it's going to generate hopefully an antigen inside the host. So that antigen can be from DNA virus an RNA virus or even bacterial. So if it works, it should work against RNA virus.
Mm-hmm.
And we're also, you know, a little bit later in this this chat, Jared Musa is going to talk about how your immune system responds to vaccines and how it's antibodies that would actually prevent the virus from entering your cells. So another question, how effective would vaccines be against preventing deaths from the acute respiratory syndrome or cytokine storms? Maybe Karen, you can address this.
Yeah, we don't know exactly the nature of what elicits that cytokine storm. We talked a little bit about this last time, but again, it's particularly a problem in these individuals that are already down this chronic inflammatory pathway, like type 2 diabetes, obesity, hypertension. Those are already inflammatory diseases. So these people are walking around with a little bit more of these inflammatory cytokines than the general public. When they get hit with the virus, we think that that's like adding fuel to the fire. And so it presumably, if the vaccine is working well, that would shut down that virus infection at a very early stage before that kind of chronic slow burn inflammatory response could go on. So that's the hope. And there are different types of immune responses, and we're learning a lot about this in COVID, and it's thought and it's hoped that a vaccine would target the immune response in a productive way to limit the virus instead of contributing to this inflammatory response. So the hope is, yes, that should be able to prevent that kind of pathological immune response.
Okay. Another question, how realistic is it to expect widespread production and availability of a vaccine in the US before the end of 2020? So that's actually kind of the next
the next slides, the next two slides might be helpful to sort of visualize that. And there are just two very quick slides that really address that. And so the first is the traditional timeline for the way we make vaccines. And on the very left-hand side, it's, you know, we target, we identify what is the target, and that takes sometimes years of research in the labs to try and identify what's the best protein in this virus or in this bacterium that would elicit an immune response. That's all basic research. Once that has been identified, then there's an effort to develop a partner that can help move that vaccine through the process. We do preclinical studies, as we said, in animal models. If those are successful, then that is presented to the FDA, and the FDA allows a phase 1 clinical trial in humans to be initiated. And that phase 1 trial is simply designed as small numbers of people to just say, is this safe? We can't, you know, do no harm. So we have to say it's safe. If that's successful, and that takes a while, then that's expanded to a phase 2 trial, which is a little bit bigger. Again, it's more safety, but also starting to say, are we developing the types of immune responses that we hope for? If that's successful, then it goes to a phase 3, which actually says, does this prevent the infection? So if we have a group of people that are immunized, do they have the infection less frequently than the group that is not immunized? And then if all of that's successful, if it's safe and effective, then it's awarded a license from the FDA. All of that at the same time requires the development of manufacturing capabilities so that every unit that is making that vaccine makes it exactly the same. Every batch is exactly the same. And that takes a lot of quality control. For a pandemic situation like we're in, which is almost unlike any other, is to do this at a much more rapid pace. So a lot of those steps are done in parallel. A lot of vaccines, as Michelle and Biao said, are in the pipeline. We don't know which is going to work. Maybe multiple ones are going to work. At the same time, this has skipped a step and gone in some cases directly into humans, while the preclinical animal safety studies are also going on at the same time. That typically doesn't happen, but it saves time. The other thing that's going on in parallel is some of the companies are making the investment, and the countries are making the investment in developing manufacturing capabilities when we don't know which vaccine is going to be the winner. So that's something that doesn't typically happen. So all of these very measured steps, as I showed in the previous slide, that is going on all simultaneously, and that's very unusual. And that will advance things in a much quicker pace. Instead of several years, it will shorten that time frame. I don't think anybody can guess at what that time frame is going to be. Is it going to be December of 2020 or not? But it's moving at a pace that is almost unlike any other, except for maybe in the days of the polio vaccine. So I'm hopeful.
Yes, I think on average, when we're not in this new world order, the average time for approval of a vaccine is 15 years. And, you know, like Karen described, you know, it's definitely accelerated in this new world order. So a couple other questions along the lines of vaccines. The question has come in, do we know if this is going to have to be a yearly vaccine or seasonal vaccine like flu, or is this going to be more like a MMR type vaccine where you get it once and you pretty much have protection for your lifespan? Yeah.
Yeah, so that's a very good question. And I hope what we can get is MMR vaccine, that you can get a vaccination and, you know, for a few years at least, you don't have to worry about it. And then so far, the virus hasn't really mutated much, and as like influenza. So that's a good sign. And from our perspective, our approach using a live viral vector and should generate long-lasting immunity and also generate relatively broad immune response. And by that, I mean, you know, you have both like antibody, you can hear more about it, but also a cellular immune response. So we are hopeful that the vaccine and that then at least the platform we are using will be the one shot you can be hopefully not to worry about for several years. And I think that's something we're really excited and working on right now.
And I'll just chime in that historically, the coronaviruses that are found in livestock animals, they have successfully developed vaccines towards those coronaviruses that show good efficacy and durability for cows and pigs. And some of that work has actually been done at the University of Georgia in Mark Jackwood's lab. Some of you may remember that he was on the first chat. Okay, one last question in this section, and then we'll move on to therapeutics. So a question came in about the fungal infections and the immune compromised folks. So the folks that are ending up having severe symptoms from COVID-19, are they already infected with these fungal infections, or do they become susceptible because of their situation on the ventilators?
Well, that's a good question, and it's difficult to tease that apart, but I showed in that one slide, there's that intermediate category of individuals that just seem to carry it around. They're okay, but they don't clear it completely. And so those individuals may be more susceptible once they have the second insult of SARS-CoV-2 and the ventilator to then develop full-blown disease. The other is, we do know, aside from COVID, that hospitalized patients, even post-surgical patients, people that are just slightly immuno off, are susceptible to these types of fungal infections. And the ventilator, because it's an invasive procedure in the lung, makes you more susceptible to getting those. But there have been outbreaks recently in Seattle Children's Hospital for one of these aspergillosis, and they were just kids that were in there for a surgical procedure. and they encountered this organism and then developed very, very severe lung infections. And so that's hard to tell. We think that certainly any of those groups of people with mild immunocompromise would potentially benefit from this type of vaccine, so that when they do encounter it in that situation, we would hope that they would be either protected or have less severe disease.
All right. So I know there's a number of antibody questions. So we're hoping that in this section of the discussion, we're going to get to those questions, and you'll have a better picture of what's going on in our bodies, both in response to seeing the virus for the first time, as well as if we were to develop a vaccine, what the response would be to a vaccine. So today, Dr. Jared Musa is going to talk to us about one branch of the immune system, which is this antibody branch, and he's going to go into some detail about isolating antibodies that prevent the virus from entering our cells, so-called neutralizing antibodies or therapeutic antibodies. And we'll also try and get at the various questions that have come in about the antibody tests that are now available through Quest and LabCorp, and now Roche has come online. So Jared Musa is an assistant professor in the Department of Infectious Diseases in the Center for Vaccines and Immunology at the College of Vet Med. His research focuses on identifying, testing monoclonal antibodies and antibody therapeutics as treatments for respiratory infections. And so I'm going to turn it over to him to guide us through this antibody section.
Great, thanks, Michelle. So this slide shows a picture of, in general, what an antibody looks like. So antibodies are proteins that are made by a particular cell type of your immune system called B cells. And they all sort of have this Y shape, and those two arms that are highlighted in green are parts of the antibody that will actually bind to and hopefully neutralize any virus that the antibody might encounter. So really the goal of vaccines are to elicit a robust immune response against whatever pathogen you're trying to protect against. And one aspect of that immune response is usually trying to elicit antibodies that can bind to and stop the virus. And you can go to the next slide, thanks. So we've heard a lot in the news lately about antibodies and vaccines. I just wanted to give a quick comparison between the two. So antibodies, again, are those molecules of your immune system that can bind and hopefully neutralize a virus, whereas a vaccine, as you've heard about today and earlier, are really pieces of the virus that can be administered to a person that will train your immune system to make antibodies. So antibodies really work by directly binding to a virus and can typically be used to prevent infection, and in some cases even be used to treat infection after symptom onset, whereas a vaccine is really used to, again, train your immune system to help produce antibodies against a virus. Antibodies can almost work immediately after they're administered, whereas a vaccine typically takes 2 weeks, sometimes even as long as 3 weeks, to elicit an immune response that is actually going to be protective. However, antibodies are only going to last a few weeks, maybe up to 6 months in some cases, because they will naturally be degraded by your body as they're circulating. Whereas a vaccine, since it's training your immune system, some vaccines, such as the MMR vaccine you've heard earlier, will protect against measles, for example, for the lifetime, or at least a number of years to prevent infection. And when we're talking about antibodies in the context of vaccination or infection, I mentioned a vaccine will take around 2 to 3 weeks to be able to protect you. And again, I'll reiterate back to like influenza. That's why it's always good to get your flu shot early, because it does take time for you to have, for that vaccine to actually work. So in the beginning, you'll have sort of these naive B cells of your immune system circulating that have never seen the virus before. Once they're exposed to a virus, those B cells will undergo a process to start generating antibody molecules. By the time that happens, typically, and you start getting sort of the best antibodies, the infection has more or less subsided. So what vaccination tries to do is to take that first part, that primary antibody response, vaccinate someone, so you turn those naive B cells into what are called memory B cells. And if you're infected again, or you're vaccinated and see the infection for the first time, those memory B cells will remember that virus and quickly generate antibodies that will protect you from infection or reduce the severity of your infection.
So Jared, let me hop in here with a couple questions that have popped up. So when folks are going to get the antibody test or the serological tests that are now available, are these tests indicative of you being protected per se from infection of SARS-CoV-2 again?
So we don't know that quite yet. It's still a little early to answer that question. We know from animal models that infection will protect against a second infection, but there's really not enough data out there so far to indicate that antibodies in particular are going to protect humans against a secondary infection. I'll say that it's my hypothesis that that's probably true, similar to other respiratory pathogens or other viral infections. But so far, we don't have enough data so far to say that. So just because you have an antibody titer does not mean, as far as we know so far, that you're going to be protected again.
Okay, that's good to know. Second question along those lines, and multiple questions actually, are which of the antibody tests are most reliable?
Yeah, that's a tough one. So there's a few tests on the market now. I think most of them are more or less fine for determining if if an individual has antibodies. If you're going through one of these commercial companies, I would have high confidence in those tests. Of course, there's many on the market from different companies. We can do some like on our research lab. In general, we're just kind of surveying the population. But most of the tests on the market are more rapid, and in general, they should be fine for determining if someone has been exposed or if they have antibodies to this virus.
Chet, any opinions?
I think that all the data coming out of the antibody tests are, as Jared said, I think that we get an idea if you have an antibody that can recognize the SARS-CoV-2 virus. In many cases, this spike protein that Bial mentioned, which is really important. And if you generate good antibodies against it, you may be protected. But I think that it's very clear that even if you have an antibody, as Dr. Musa said, it does not mean that you are protected. So if you get a positive result on that test, it does not mean you should feel like you're at lower risk or anything of that from a reinfection. With regards to the actual tests out on the market, just like we've talked about in the previous sessions, I think that a lot of things have been released under an emergency use authorization by the FDA. And that is because we needed something quickly. And a lot of these tests have not gone through some of the very rigorous testing that, say, a glucose test or a cholesterol test has, that really has defined precision and defined accuracy. So for example, we don't know with some of these antibody tests, as we're seeing news outlets and data emerge about false positives as well as false negatives. We don't really know kind of what's the role of other coronaviruses that cause, for example, the common cold, that may also induce antibodies against this virus. But as Dr. Musa said, they don't protect you. So there's a lot of caveats associated with that. And really what we need, and we talked about this last week, is a high throughput way to say that someone has generated antibodies that are protective and that those antibodies are present in their sera. Simply relying in their bloodstream, relying on does something bind or not, as we also talked about, is not, or interact with that virus, isn't exactly good enough. So, you know, and in terms of which test is best, I think that they all have pros and cons. You know, I see some nice data from the Roche test, but now as these come online, they're going to go through more rigorous review by research labs like scientists as well as the FDA. And I think that you will see the kind of a shift towards what test is the best, makes the most sense, and may in the end have some, you know, predictive effect. But I would caution people largely on those tests up front.
And so, yeah. Jared, another question has come in on serological surveys in Athens, and I think your lab is involved in some of these analyzing these surveys. Is that right?
Yeah, so we started pretty early on looking back, because a pretty routine part of our research is we collect blood for some of our other projects, and we have all those samples in our freezer. So we just looked back, and luckily we haven't found really any positive people as far back, you know, pretty much from December and before that. And now what we're doing now is we just have our protocol approved to start recruiting patients who have tested positive for the coronavirus to, as I'll talk about in these next couple slides, to actually look at their monoclonal antibodies and see if these antibodies could be used as therapeutic treatments.
Okay. All right, so I'm going to let you get on with your slides, and we'll come back to the questions and closer to the end of your presentation.
Sure. Yeah, so I just wanted to show what we talk about the spike protein or viral proteins in general. So on the left there is just a cartoon of the virus, and you see that red protein kind of decorating the surface of the virus. And if we look more closely now, we have structures of that particular spike protein. So the second image there on the slide is what the spike protein looks like. And then what happens is that spike protein will slightly open up a little bit. So it's sort of in this closed conformation. It'll slightly open up when it's approaching a cell to be able to attach to the cell and eventually gain entry. So as that spike proteins open up, it'll bind to your cell, and the particular protein on that cell is called the ACE2 receptor, which is pretty abundant in the respiratory tract. So really the goal is to develop a vaccine or make an antibody that can prevent that interaction from occurring. And then if we go to the next slide, so again, on the left there is showing that spike attached to that ACE2 receptor, and on the middle is what sort of our goal is. So we want an antibody. So I have the image there on the right showing what, again, an antibody looks like. So that single part of the antibody highlighted in green, we really want that to be able to bind to the spike protein and sort of mimic and block the ACE2 protein that's present on your cells. And if we can if we can make an antibody that could prevent the virus from even binding to the cells, that's sort of the optimal goal. So the virus would enter your system, you'd have antibodies around that would sort of coat the virus right away, block it from even attaching to your cells, and then the rest of your immune system would be able to clear it. And then if we go to the next slide, so what my lab is working on, I mentioned we're doing sort of these serology studies. So we're looking back before this pandemic to see, do we have any antibodies that might protect against this virus, or at least bind the virus. So is our immune system sort of like halfway there, or are we really at the very beginning when we get infected? And we're making monoclonal antibodies against the spike protein before people were infected. So again, the sort of pre-existing antibodies, and then also after they're infected. And we're also looking at some of the other proteins in case we find out that really the spike protein was not the best vaccine candidate out there. Although I expect it will be, but we're also looking at the membrane protein shown in the cartoon there on the right, and also the envelope protein. And we know, for example, from SARS from 2003, which is another closely related coronavirus, that antibodies to the membrane protein did elicit neutralizing antibodies. And then for the envelope protein, we know similar proteins are present, for example, in influenza virus, and that antibodies to that protein will protect against infection. So our lab is sort of taking this multi-pronged approach focused on antibodies to try and target this virus at multiple angles, as well as looking at the human population here in Athens to see if people, how many people are infected, and recruiting people who might have previously been infected as well. And it's sort of a complicated process, but basically we will draw blood from people at the, here at the university at the Clinical Translational Research Unit. So they've been really great working with us for a number of years. So we'll have blood draws there. We'll drive over to the health science campus, take the blood back to our lab. And we can actually identify those specific cells in your immune system that will bind the virus. And using a number of different techniques, we can identify those cells. We can take that cell, a single cell, and identify whatever antibody it's making. And then we can take that information that we discover from that antibody and then take that gene and make it in our lab. And then once we have that antibody made in a different cell line, we can start testing it, whether it's to see if it can actually neutralize the virus. And I think I will stop there. Of course, I can take questions.
And so, Jared, conceivably, since you can sequence antibodies and you have assays to determine if they're actually neutralizing antibodies, can you then synthetically make a therapeutic antibody because you know the sequence?
Yeah, absolutely. That's really the goal here. So we'd be able to identify whatever that sequence was from a person who was infected, scale it up, hopefully through partnerships with industry, and then begin testing that antibody in animal models. And I will mention that UGA, especially in our college, we have really excellent facilities for doing all of this research. And we'd be able to take that antibody and hopefully get it into people as soon as possible for testing.
Yes, and I'll also chime in and piggyback off of the statement that the college has a lot of abilities to test not just therapeutic antibodies, but to also test vaccines that are being developed by our faculty, as well as vaccines that are being developed at other companies, as well as other academic institutions. So we have unique resources to be able to do that, and we have unique animal models to be able to test interventions for other people as well. So a couple questions have come in about coronaviruses and whether or not they mutate like flu. So I'm going to open this up to the panel as well. There's been a lot of titles of papers that make it into mainstream media that suggest that coronaviruses are mutating and becoming more virulent, and that that's going to cause problems in vaccine development. Most of these papers have not been peer reviewed. and most of the papers don't actually report data to support those sensational headlines. So maybe we can start with Biao and talk a little bit about what viruses do, which is mutate.
Yes. So change is constant for viruses, and that's the way of life for them. So they're going to change. And fortunately for us, the coronavirus we have right now, and it's in the beta coronavirus, they haven't changed that dramatically, make them so different. If you look at all the isolates from different places, and they seem to be fairly similar. So in that sense, I think we're very fortunate. If we do develop a vaccine, again, we should be able to contain this infection pretty well. And will they change in the future? Something new will emerge. I say there is a high probability a new kind of a virus will emerge. And that's why as a scientist, I find this is interesting and to some degree exciting. I realize for society, this is terrible and we have to be prepared for it. But all the things we are doing right now, you hear from Jerry's antibody in isolation and Karen's vaccine to target, comorbidity factor and et cetera, all the things, and we are developing a platform system that all the things we're doing can be readily applied for a new and emerging coronavirus or any other emerging viruses. And I think that's one of the things we hope you walk away and knowing that we are working on this kind of things, not necessarily precise the same thing. Like for instance, we've been working on MERS for 5 years, for past 5 years. There are, besides us, there are quite a few labs, and not as many as we like, working on this. And so again, MERS has not turned into SARS-2 and COVID-19, and they've been circulating. They're still circulating in the Middle East. So yes, they will change, but they are not fundamentally changed into a new thing. And what worries me, to be honest with you, is some kind of new virus is going to emerge from somewhere. We don't know. And the best we can do is be prepared.
So Jared, I'll throw it back over to you since you work on flu as well. So the fact that flu mutates and we have to get a different vaccine every year, how is flu mutating or drifting different from what we're seeing with coronaviruses?
Sure, thanks. So it's a little bit different for influenza than for this new coronavirus. Like Dr. He said, viruses mutate. Some viruses mutate more rapidly than others. For example, influenza mutates quite rapidly, which is why we need a vaccine, a new vaccine, almost every year. And for the coronavirus, though, influenza has specific mechanisms to mutate based on the way its genome is set up. For the coronavirus, that's not the case. So while we will see small mutations in the coronavirus over time as it's circulating in humans, it's very likely that those changes will not affect the virus pathogenicity, and very likely that the vaccine that's being developed now will still protect against the viruses that are going to be circulating. Hopefully not, but if they are still circulating when we get to the point where we have a vaccine on the market, those vaccines will most likely and hopefully still protect against what's circulating out there.
And so I'll also point out that coronaviruses, unlike flu, have a proofreading mechanism. So when mutations are introduced into the virus as it replicates, it has a mechanism to correct those mutations, whereas flu and many other viruses don't have that mechanism.
Yeah.
Jared, there's also another question related to the serological study. So how can folks get involved in that study, or can they be involved in that study?
Oh, sure. Yeah. So people need to contact the UGA Clinical Translational Research Unit. So they handle all the recruitment and everything for those studies.
And then one last question before we move on from antibodies. There was a question that came in about using convalescent plasma, and what are the effects of using this plasma that gets transfused from individuals who survived in the infection. So does this actually activate naive B cells, or patients that have been naive to the virus previously? And what's the outcome as a potential treatment? So I'll open that up to both Jared and Chet.
Yeah, I can just start. So that transferring serum is actually pretty old technology, maybe 100 years old. And basically it's just taking serum, so the clear part of your blood, and transferring it, or plasma, and transferring it from one person who was previously infected. So their plasma is going to contain antibodies to the virus. So those antibodies can basically be transferred to another person to either prevent them from getting infected, or to help treat somebody who's already infected. That procedure is very safe. As I said, it's very old technology, and it's been used for a number of different pathogens, and it's been used for well over 100 years now. If it's going to work for this new virus, we don't know the answer to that yet. There's a number of clinical trials going on to ask that question. And I think that's a promising method to help treat those who might already be infected, or to help prevent those from getting infected, like frontline workers in the hospital, for example. And that again is sort of the bridge until we get good monoclonal antibodies on the market that are much more reproducible, and hopefully we'll be able to prevent and treat people as well.
Great. All right. So I'm just gonna do a time check. It's 2:02. These chats have typically been going about 90 minutes in length, with all of the questions, which we're really trying to get to. We have two more topics that I'm sure everybody's going to be familiar with from news coverage. We're going to move next to hydroxychloroquine, and Chet's going to talk to us a little bit about how hydroxychloroquine works, and what it was originally developed for, and what are we seeing in well-controlled clinical trials. So as many of you know, Chet's been on all three of the chats. I'll just reintroduce him for those folks that are new. He's an assistant professor in the Department of Infectious Disease and the Center for Vaccines and Immunology in the College of Vet Med. His research is primarily focused on the role of antibodies and B cells during infection and vaccination. His specialty is malaria, which is why he's going to be talking to you about hydroxychloroquine. And he's predominantly interested in the impact of antibody responses against malaria, and also co-infections of things like Ebola. And he's been using his expertise in these areas to try and understand the COVID-19 immune response, and how antibodies might be able to protect against the infection, as well as potentially if they could contribute to the disease. So I will let Chet give you guys an update on hydroxychloroquine.
Thanks, Michelle. So I think that before COVID-19, people that knew about hydroxychloroquine were not that many. And now everyone knows about hydroxychloroquine or chloroquine. And so what I wanted to just talk to you about today is, what is this drug? How does it work in different situations that it's prescribed for, and in situations that it's not necessarily prescribed for? And then give you a little bit of insight into the big picture view of how the clinical trials and the observational studies that were done related to this drug and COVID-19, where we kind of sit now with everything that we know after doing those well-controlled trials. So hydroxychloroquine is a derivative of the drug chloroquine. It's called hydroxychloroquine because it's been altered a little bit compared to chloroquine. And what this translates into is just a little bit of less toxicity and side effects. But essentially they do the same thing. And that is that they primarily will kill malaria parasites. That's definitely what it's most known for. And it also, and some of your relatives, if they have autoimmune diseases like lupus, is known to modulate your immune response and prevent that autoimmune reaction and relieve some of the symptoms of lupus. So this drug is marketed under the generic name hydroxychloroquine sulfate. And then you may see it under the actual trade name of Plaquenil. This is what a lot of people take that have, for example, the autoimmune diseases. And as I said, I think it's a beautiful picture, very close to my heart. You can see in the bottom left, malaria parasites actually infected red blood cells. And the red little pink dots with nothing in the center that's blue, those are just normal red blood cells that you would find in your bloodstream. And the ones that have the blue and some shades of pink and that really beautiful coloration inside of them, those are the ones that are actually infected with malaria parasites. And what this drug would do if it was given to someone with those parasites is it would kill them and have them cleared. The way that this drug works in a variety of diseases or in malaria is it actually interferes with the ability of the parasite to eat its food when it's inside the red blood cell. So inside your red blood cells, the parasite eats hemoglobin, which is just a protein that's found in red cells that helps carry the oxygen throughout your body. So this is what oxygen binds to and then gets filtered through all the different organs and things in your body. What happens with this, and when you take chloroquine, is that beautiful little circle with those crystals inside of it, that's the parasite's stomach, if you will, its little food vacuole. And that whole process is very complicated when you're breaking it down, but ultimately this drug prevents that breakdown and makes their own food toxic to them. And it causes the parasite to arrest and die. If you go to the other thing it's used for is it can actually tamper down or disrupt an immune response against yourself. So last week we talked about the concept that your immune system's role is really to say, I don't recognize something, I need to get rid of it because it may cause me harm. And that is termed as self versus non-self to us immunologists. And the way this works is in autoimmune diseases, your body starts seeing things that are you as foreign and says, we got to get rid of that. And so in diseases like lupus, rheumatoid arthritis, etc., people prescribe antimalarials because they actually can alter some of the molecules that your immune system sees and reacts to, so that your immune system no longer activates and no longer fights against those molecules, and thus you get relief from your symptoms of that disease. And so that is one way that this works in autoimmune diseases. And again, it's working through very similar mechanisms by essentially messing up some things that we'll talk about now. With viruses, it's been known for quite some time that in cell culture systems, so not in animal models or in people, but at least in culture, when you only have a cell and the virus, not the immune system and not all these other pieces of the puzzle, when you actually graduate into humans, it's been known that this can interrupt the virus when it gets into that cell and it's unpackaging and how it goes through making more copies of itself. And it can hit multiple parts of that process. But essentially the way it's thought to work is that just like we have a stomach, cells have a stomach as well. They have something that has a really really low acidity that helps them digest things apart. And the virus has to go through that whole process. And this drug prevents the acidity from happening in that process. And thus the virus can't get out and it can't make copies of itself. And so in cell culture, it clogs up that system and you don't get more virus production. So all of that information from the perspective that in malaria, this drug is pretty well tolerated. I've taken it. It's not horrible. It's not great either, but it's not horrible. And people take it for lupus and RA long-term with no issues. There was kind of a rationale to see, since there was some early data that said in cell culture, this also prevents SARS-CoV-2 from making copies of itself to progress into the clinical trials. Now, I want to highlight that just because it happens in cell culture, again, and this doesn't only apply to hydroxychloroquine. I know that a lot of people are seeing in the news various extracts of mushrooms, extracts of various plants that actually prevent the virus from growing in vitro or in that cell culture system, that maybe it has some effect. That is very possible that there's something in that plant or something that causes in that cell culture system without your body, to prevent the virus from growing, but it doesn't mean that it would work in a person or human, and it actually may be toxic. So as Karen pointed out, just like we have all these checks that happen for a vaccine, we have similar checks that happens for drugs. And I want to just be clear that just because it works in cell culture, it doesn't necessarily make it into humans. And we're not advocating to try a lot of these things. Anyway, so there was a rationale to move into clinical trials, and there was some very early data. It made its way around social media. That's actually how I encountered it myself the first time. Out of France, it suggested that if you took hydroxychloroquine with or without a Z-Pak or zithromycin, you may actually have less virus at the same time as people who did not take those drugs. That was encouraging. It was definitely at a time when we had nothing and people were really nervous and trying to find something that they could do to help people. However, there were a lot of issues with that study. Some of them was that it wasn't a double-blind placebo-controlled trial. So that means that the doctors knew what they were giving, and there was no real true placebo arm where patients were also not given the drug. And that makes it complicated because, as we all have heard before, placebo effect happens, and we have to control for that when we're looking at drugs. Their control groups or their comparisons to where they were saying virus was actually being cleared earlier was not great. It was not in the same facility. It wasn't necessarily with the same enrollment criteria or people they include, and that made it hard to interpret. And importantly, they didn't actually talk about issues with patient care in that preprint manuscript, and they never did when it was actually released. And actually, if you read the fine print, there was some evidence that one of the complications that we know happens with chloroquine or hydroxychloroquine, which is heart issues, can arise in a subgroup of individuals, especially when you give the drug at high concentrations like was being done in this particular situation. In fact, it's about 3 to 4 times higher than what you would give for a malaria parasite infection. That there might have actually been worse outcomes. Nonetheless, the world went forward with trials, properly controlled, large trials with a lot of people in a lot of different places throughout the world. And one of those studies that was published in The Lancet has suggested there's really no benefit to treating people with hydroxychloroquine that have COVID-19 or have disease, and it may actually cause harm in the form of this heart abnormality known as an arrhythmia. And if you look here on the bottom right, this is just some data from that trial. If you have a mark on the right-hand side of the center line, it means you were more likely to have a higher risk of death compared to a control. And on the left-hand side, you have a decreased risk of death. And as you can see in the bottom right in the red, all of the drugs given with or without what we call macrolides, which really isn't important for this conversation, you actually had a little bit a higher risk of actually succumbing to the disease. And so this is important because I think that this highlights the essentially what we have to do and some of the consequences if we're too quick and we make too rash decisions. I think that we can, have some detrimental consequences. But hopefully this gives everyone an idea of what hydroxychloroquine, where it came from, what's it been used for, and also kind of where we sit now. I've not seen any data, and if the panelists have, I would love to hear it, where we've actually looked at, if you take it early, does it prevent you from getting infected? I don't know if we've answered that question yet. I do know for a fact there are clinical trials asking that question, but certainly if you have the virus, there's actually an increased risk of problems. So.
Great, thank you, Chet. That's pretty informative. So we're going to move to our last therapeutic intervention. We were supposed to be joined today by Dr. Jeff Hogan, but he wasn't able to make the talk for a last-minute meeting he was pulled into. So I'm actually going to talk to you a little bit about the antiviral drugs that I'm sure are top of mind for folks, especially a particular drug called remdesivir. From this schematic, which I borrowed, I thought it actually presented very well for the life cycle of coronaviruses and other viruses as well. And so essentially, you'll start with your virus encased, the genome is encased, it has receptors, or the spike protein, are these little lollipops, and this is your epithelial cell, if you will, lining your respiratory tract, and these are the ACE2 receptors. So you get binding of the spike protein to the ACE2 receptor, and in theory, if Jared's successful and we have a therapeutic antibody, this is where that therapeutic antibody would block. It would block the binding of the spike protein to the ACE2 receptors so that the virus could not get inside the cell. The virus needs to get into the cell, and it uses the cell's machinery to replicate its genome. And so you have binding on the outside, you have kind of invagination or absorption into the cell. These would be your lung cells. And then you have release of the viral genome. And in order for this virus to survive in you and be shed out and transmitted to other people and replicate in other people, it has to reproduce its RNA genome. And so it uses your own cell's machinery. So this is machinery that your cells use to reproduce its own genome. So it takes advantage. And then it produces new genome and new viral protein to protect it so that it can then go off into the world and infect others. And that's how viruses live. It's kind of a symbiotic relationship, because if they kill their host, they kill themselves, and they can't replicate outside of their host, whether it's a bird or a bat or a human. So for other viruses that we know about, companies have actually developed small molecules that prevent various aspects of this replication. And so one of the ways to prevent this replication is to use antivirals that take advantage of the building blocks that are required to build the viral genome and your genome as well. So in the sense of an RNA virus, you use the building blocks are uracil, guanine, adenine, and cytosine. In the case of DNA, you don't have uracil, you have thymine instead. But we're talking about RNA viruses. So there's machinery, your own cell's machinery, that will sit along this parent strand, and it will build a new genome strand that then gets packaged into a new viral particle and goes out into the world. The nucleotides or nucleosides or building blocks that your body uses are made up of a base, so one of these bases, and then a sugar gets attached, and then you have your final nucleotide, and that becomes the building block of your genome. So what chemists have done is, in the case of remdesivir, they have taken adenosine, so your sugar molecule, your base, and they made a chemical modification to your building block. So it's essentially a fake genetic building block, but it mucks things up for the viral replication, because when this modified building block gets added into its spot in the genome, the machinery that will continue along and continue adding bases, it stops because of this one modification. And so you cannot continue to build the whole genome. And you stop viral replication. So that's one mechanism for how you can inhibit viral replication. And the idea would be that you could give this drug early when you test positive with the PCR, the molecular test for COVID-19. The problem is that remdesivir and other drugs like it are not in a pill format. So they actually have to be delivered IV over 5 days, and so you have to be in a hospital setting. So some of the exciting work that's being done at UGA is to try and identify more of these fake genetic building blocks and to be able to formulate them so that you could actually take them in a pill format. And so this is kind of an old idea as well, because cancer chemotherapeutics also take advantage of this type of mechanism. So viral genomes need to replicate very quickly, and cancer cells have to replicate very quickly. And so if you introduce these fake building blocks in both settings, it quickly shuts down the replication. But you do introduce toxicity because your body will also try and use these fake building blocks in its normal process of replicating its genome. And so there are definitely toxicity issues with this type of mechanism. There's another mechanism. So I talked about viral proteins, like encasing the genome so that the virus can go off and be transmitted and infect other people. And so we also have what are called proteases. So protein enzymes, or enzymes that degrade proteins. And at UGA, we're looking to identify very specific protein enzymes that will degrade the SARS-CoV-2 viral proteins. And this will also inhibit viral replication. So you have much less virus for your body to clear, with the idea that you will have much less of the cytokine immune response, but you'll still develop that B cell immune response and hopefully some neutralizing antibodies from seeing at least a little bit of the virus. So again, two different mechanisms, small molecules that we're actually trying to hunt for in laboratories at UGA. So I'm actually going to stop there as far as therapeutics and vaccines. I'm going to go back to one question that we really didn't address, which I think is going to promote significant discussion, and that is herd immunity. So we've talked a lot about antibody response in infected individuals. We've talked a lot about vaccines. And so I'm going to open it up to the entire panel to talk about what does herd immunity mean, and what do we have to, how do we get to herd immunity? So maybe Karen, you can start off.
Yeah, herd immunity refers to the concept that a large percentage of the population, the herd, has protective antibodies, and that when they encounter a virus or an infective agent, they would be protected from that. And if enough of the people have that protection, then the ability to sustain the transmission of that virus, for example, falls off because enough people are protected. And the benefit of that is particularly important for the people in our population that would be, again, these highly susceptible individuals, and that have a tougher time developing immunity to the natural infection or to the vaccine. And so herd immunity can develop by natural spread of the virus. We all make an immune response and maybe we will be protected, and that will dampen the transmission. Or more ideally, if we could just vaccinate everybody or a large percentage of the population and they would develop a robust immune response, then those individuals, even if they're not vaccinated or cannot be vaccinated or cannot develop an immune response, they benefit and are protected too, because the rest of us are immune and the transmission rate goes very, very low. So it's less likely that even the people that are not developing antibody responses can be protected. So the herd is protecting everybody. The difficulty here in the COVID situation is, as far as we can tell right now, there's still actually a low enough percentage of the population that even has antibodies. And as Jared said, we don't even know if those antibodies are protective and preventing the infection, the spread yet. We don't know just the basic immunology and biology of the infection yet. So we can't rely on herd immunity to protect the larger population. The other problem is the individuals that do get this infection that require hospitalization, it's actually very, very severe. So most people do okay. They come across the virus, they do okay. The individuals that are susceptible and that would benefit from herd immunity, those are very, very difficult infections. And so we can develop herd immunity eventually if we all see, or a large percentage of the population see the virus and that natural immune response is protective. We don't know that yet. The other way to do it is to vaccinate, where you can vaccinate 70 or 80% of the population, and then those unvaccinated individuals, either very young or very old or people that are susceptible, would benefit. So it is more likely to be the result of a larger vaccination strategy than eventually all of us getting infection.
And so is it fair to say that the social distancing measures, the wearing the mask measures, these are really measures to delay and give us time to develop a vaccine, give us time to understand how to treat patients in the hospital that are having these severe symptoms?
Right. We can't rely on just transmission to provide protection to the herd. It's too deadly of an infection. It's too deadly. So social distancing, wearing a mask, washing your hands, those are the measures that will keep the transmission down. And people have studied this already and said, yes, in fact, if a large percentage of the population wore masks and adhered to social distancing, the transmission would fall low enough that people that are really at risk would benefit from that. All of that should be going on while in parallel, we're working very hard to develop a vaccine or better treatments, okay? But right now, the low-tech strategy is social distancing, wearing a mask to protect other people from you, primarily, and washing your hands, good hygiene, quit touching the face. All of those things, those are low-tech science things that have been proven to be associated with lower transmission. The higher tech things that we're talking about take time, money, thought, and good luck.
Yes. All right, so I'm gonna open up the final question. I will say that this is probably gonna be a geek out question, and it's predominantly directed towards the antibody panelists. So there's been some interesting work with the use of IgY antibodies to neutralize a prominent allergen in felines at the source, which is apparently in the mouth of cats. Could we see something similar to this for a virus like COVID-19, maybe at the point of entry into the respiratory tract? So I'm going to open this to Biao, Jared, and Chet, because I've never heard of IgY.
Yeah, the IgYs are antibodies from chickens, and they're actually present in eggs. So you can engineer chickens to produce, anti-coronavirus antibodies. I don't know how effective they would be. In that case, you would have to eat them, I would guess. That would be the method of delivery, which would probably work great for stomach virus. I'm not sure how well it would work for a respiratory virus, but maybe the other panelists can add on to that.
Yeah, so I think that this is touching on an interesting topic, which is that in biotech, there's actually a theme, even the father, if you will, of modern antibodies, who discovered antibodies, Max Cooper, who's at Emory, actually looks at this weird fish that lives in mud and is quite terrifying called a hagfish. And he has the company generating their form of an antibody and using it to neutralize different pathogens. But people have also looked at camelid antibodies, so antibodies from camels, and also shark antibodies have also come up in this realm. And now these these IgY antibodies that Jared mentioned. And I think the attraction is that there's some features of those antibodies, like that they only have one subunit. So they don't have those multiple colors that we talked about. So they only have one subunit, and that makes them a little smaller. And so maybe they will penetrate certain parts of your body easier than, say, others, like the big human antibodies that like Jared generates or from other animal models like mammals. But I think what's interesting to me about them is we really don't know enough yet about how the antibodies really truly regulate this infection to dampen the host response versus how do they actually mediate the protection against the virus, because antibodies work in many different ways. And so some of these antibodies from other species may not be able to interact with our immune system in the same way. So I think there's a lot of work that we want to do here at UGA and the college, asking some of those basic questions that may inform if this type of approach is going to be a reasonable approach, or maybe not. But I think it's a really cool area. It's like, as Michelle said, a geek out moment of this weird, how the immune system has evolved and essentially ended up at the same place to do the same function, but with different flavors, different ways of accomplishing that goal.
I think the question is really, really interesting. I think the person asked is really on the right track. That is, if you think about the coronavirus we have, it's a respiratory pathogen. And most of the antibody, if you hear about it, are focused on IgG, right? So actually, as I mentioned earlier, one of the really nice feature of the vaccine we develop is using an intranasal route. So one of the things with intranasal route is that you develop mucosal immunity. So in that case, you were expecting to generate IgA, so a little bit different. So essentially, the question is, like, can we fight the virus at the very site when it gets an entry? Say if you're in your nasal cavity or in mucosal surface when you first encounter. So those are the antibody a little bit different than the ones that in blood. And hopefully, in the vaccine, will generate enough of this the IgA that we can prevent infection from really going out of hand, as Karen mentioned early. not going down to the route of cytokine storm, etc. We're working on.
All right. Great. Yes. So buying some time for our researchers and scientists to learn more about this virus, as well as how we react to it, and developing therapeutics and vaccines. So we are a little bit over. I'm going to go ahead and sum up where we are at the College of Vet Med as far as research programs on the vaccine and therapeutic side. We have multiple vaccine discovery programs underway. You heard about Bial's. I mentioned a little bit about Ted Ross, and you heard about Karen's vaccine program. We also are trying to identify therapeutic or so-called neutralizing antibodies that inhibit SARS-CoV-2 as a way to modulate the disease progression. And then we're also testing various antivirals, drugs that are already available, such as remdesivir, trying to understand the mechanism for how this drug is actually working for SARS-CoV-2. And then, as I said earlier, screening for new antiviral drugs that could be used to treat the infection and prevent viral replication. And so we've had 3 of these fireside chats, and I just wanted to kind of summarize the global work that's being done in the College of Vet Med. So we're investigating everything from new tests, new reagents and tests to alleviate some of the roadblocks for being able to have more testing here in the state of Georgia. We're trying to better understand how this virus is transmitted. We're looking at the underlying immunological response for folks that are seeing this virus, because everyone's seeing it for the first time. We're trying to understand, why are certain patient populations suffering more severe symptoms than other patient populations? Why are folks developing blood clots 3 weeks after being infected when they have a low viral load? Why are kids a month after being infected developing the Kawasaki-like or toxic shock syndrome, and you're 4 weeks after seeing the virus and you've mounted an immune response? And, why are folks that are diabetic or are more elderly in age or have issues with obesity, why are they in such high populations being admitted to hospitals and just not having a positive outcome? And then lastly, multiple laboratories trying to develop vaccines and therapeutics using multiple different approaches, so we don't have a one-size-fits-all in our response. And collectively, all of this information will be able to be used in the future for understanding viruses that will emerge from animals in the future. Just like, SARS-CoV-1 emerged in the 2003 timeframe, MERS coronavirus emerged later. Now we're 10 years down the road and SARS-CoV-2 has emerged. There are plenty of bat viruses that we don't know about and have the possibility of emerging. And so really developing an understanding in all 3 of these areas allows for a better ramp up when new viruses do emerge. And then lastly, I'll leave you with, we talked a bit about buying time so that our scientists have time to understand what's going on with these viruses and develop therapeutic and vaccine interventions. So I'll leave you with the tried and true public health measures. Wash your hands, cover your mouth and nose when you cough and sneeze, do your best to physical distance, wear masks. If you're sick, isolate yourself. And flu season will be upon us in the fall, so get your flu shot, because it's probably not going to be good if you develop COVID-19 along with a flu infection. And so I'll end by thanking our panelists for joining us for this last Fireside Chat, thanking the College of Vet Med for supporting these educational and hopefully informational chats, and allowing us to share with you all the research that's going on in the College of Vet Med. And hopefully we'll be able to have an extended portion of this series and actually talk with individual investigators in more depth. And so I'll leave you with that. Have a good evening, and thank you again for joining us.